Hi everyone, this is Mr. Burda here. Today for AP Biology, we're going to be talking about the Unit 8, Topic 1, which is gene regulation. All organisms need to be able to regulate their genes, whether they're a simple little bacterial cell or one of us, a eukaryotic cell. I start with regulating gene expression and gene regulation by talking about feedback loops. Feedback loops are a way to control when or when not a gene is active. Typically, when we look at feedback loops, you're looking at a negative feedback loop. You can see here in feedback inhibition. Your end product is going to come back to the beginning of the cycle, and it's going to shut that whole pathway down. So in this case, tryptophan is being produced via several genes. And once tryptophan is being produced, there's really no reason for us to continue to produce it. That would be a waste of time and effort. So tryptophan is going to come back to the beginning enzyme in the pathway, shut it down, and we stop making tryptophan. And so we don't have excessive waste in the cell. But we can also change gene regulation, which is actually the whole point of this topic. What if instead of tryptophan turning off an enzyme, what if tryptophan came back and inactivated a gene? If you don't make a gene, you don't make the enzyme. If you don't make the enzyme, you don't make tryptophan. It's just another way that feedback loops can be used to control how much waste we have. Continuing on, this is going to be probably one of the main topics here. These are called operons, which you can see right here. Operons are found in bacterial cells, and this is how they control when or when not they actually code for a protein. If you look at these sequences of genes here, these are the genes that are coding for lactose, or lactase, shall I say. The operator region, that's going to come in handy right there when we're going to look at that. We also need to take a look at the promoter region. And later on, we'll talk about what the regulatory gene does. The operator is going to control if the RNA polymerase can actually read the genes. Take a second to think, what the RNA polymerase actually does. Think about it. If you guess the RNA polymerase makes mRNA, you would be correct. The operator region is going to be a region that blocks the RNA polymerase from reading those genes. If it can't read the genes, it never makes mRNA. The promoter region, right here, is where the RNA polymerase actually binds. So the RNA polymerase is going to sit right here, and as long as nothing is blocking the RNA polymerase here, it's going to be able to read these genes and create the protein. So we have two types of operons. We have the repressible and we have the inducible operon. I'll start with the inducible. It's normally, this gene is always turned off. So it's always turned off, but can be activated. So this is usually a catabolic operon. That means something where you're breaking down food molecules. And let's take a look at what this looks like. So here you can see a lac operon. We have the RNA polymerase right here. Currently, this RNA polymerase is being blocked. Where is it being blocked? It's being blocked right here. We need to remove that. That's called the repressor. So how do we do that? Well, it's actually quite easy. What we have is a lactose molecule. Lactose is what we're going to call the inducer. Right now, I'm not eating lactose, so why would I make a gene to break lactose down? But let's say I chug a glass of milk. Now all of a sudden I have a lot of lactose in my body. What lactose will do, it will bind to the repressor. The repressor will then fall off, and the RNA polymerase can then code for the gene. The gene will be turned on. So, long story short, if I'm not drinking lactose, there's no reason for me to make lactase. However, if I drink lactose in the form of dairy, milk, ice cream, lactose will enter my body, it will bind to the repressor will fall off, and we get to code for the gene. Makes sense, it's all a way to regulate. Let's go over to the opposite. This is called the repressible operon. This one is normally on. It's usually for building. So we would call this an anabolic operon. The example that I'm going to use is called tryptophan. And tryptophan is going to act as a co-repressor. So again, this is just the reverse of the inducible operon. So here you can see the RNA polymerase is working. And if you check that out, it's moving down the stream and it's creating the messenger RNA here. So it's on. So what happens once I have enough tryptophan? Well, once I have enough tryptophan's going to come back around and it's going to bind to the repressor. Now you can see the repressor is changing shape. Now that the shape has changed in the repressor, it's able to bind to the operator and turn this gene off. So what's the point? If I have now made enough tryptophan, my job is over. This little molecule here is what we're after. Once we've made enough of it, why would I keep making more? That would be a waste of time and resources. So our end product is going to bind to the repressor, thus shutting off the genes that create it. It's a very clever way or clever mechanism of regulating genes. The second step, we're going to talk about that more so in class. How do we control our genes? I'm going to have a part 2 of that. We have a few more steps that we'll go over. But just to recap, we talked about inducible and repressible operons, how they're used in prokaryotic cells, and we also talked about feedback loops, how feedback loops are typically negative and they'll come back, the end product, shut down the pathway, then we're not wasting materials. And that's all for part 1 of my gene regulation. I'll come back with eukaryotic gene regulation.